The topic, I'll introduce myself. I'm Nitya Jain, Assistant Professor, Department of Microbiology, SN College, Alathur. Now we'll look into the mechanism of tryptophan operon. It is a negative repressible operon. This operon, which consists of a promoter, operator region and a leader sequence and structural genes. In the structural genes, there are 5 structural genes. We have only 3 structural genes in the lac operon, but here you have 5 structural genes. That is tryp, tryp D, tryp C, tryp B and tryp A. They are all involved in the various stages of tryptophan synthesis from chorusmic acid to tryptophan. So each structural gene code for each enzyme here. You can look at the tryp E, which is involved in anthranilate synthase enzyme synthesis. Tryp D, which have role in the synthesis of phosphoribosal transferase, and the tryp C involved in indole 3-glycerol phosphate synthesis, and tryp B and tryp B, which are involved in tryptophan synthase. Tryp B have tryptophan synthase beta, are involved in tryptophan synthase beta production, and tryp E in tryptophan synthase alpha production. These enzymes are very much important in the tryptophan synthesis. Now we can see how this operon is look like. You can see that there's operonic region with a promoter and an operator to which the regulatory protein is going to bind, and a leader sequence or 5-UTR region, 5- untranslated region. This is very much important in the tryptophan operon, and tryp E and the other structural genes, and you can see the structural genes, 5 more structural genes, tryp E, tryp T, tryp C, tryp B and tryp A. These are very important in the operonic region of tryptophan. And also other factor which is away from the operonic region, but they can influence the transcription of tryptophan is its regulatory region. There it is producing the regulatory, it is a negative repressible operon, so they are producing a repressor. If the repressor actually they are producing an inactive repressor. The repressor will be active only in the presence of co-repressor here. The co-repressor here is the tryptophan itself, the product itself. If the cell having high amount of tryptophan, the tryptophan will come and join with the tryp repressor, and it will get activated, and they will go and bind with the operator region. And if the amount of tryptophan in the cell is very high, there is no need to transcribe the structural gene. So they will inhibit the tryptophan operon. If it is high amount, high TRP, the repressor will active and they will come and join with it. If it is low, then what happens? There is no or that much amount of tryptophan will not be available in the cell. So they cannot go and bind with the tryp repressor, and they cannot inhibit the operator region. See, we can study the importance of leader sequence in the coming slide, but this also involved in the attenuation and anti-attenuation process of tryptophan operon. That means that can involve in the tryptophan operon effect that can repress or enhance the operon. That we can study in coming slides. What is 5-UTR or leader sequence? Actually, apart from lac operon, there is 5-UTR region. After promoter and operator, there is a 5-UTR region. This 5' UTR region, which consists of 162 nucleotide and 4 region, you can see the region 1 here, region 2 here, region 3 here, region 4 here, and a lot of uracil nucleotides at the end of this UTR region. So in the first region you can see 2 consecutive tryptophan codons. This is 2 trip codons actually. This nucleotide will transcribe and translate it to mRNA. When this mRNA comes, the mRNA will have different type of looping arrangement. If the looping arrangement, the secondary structure comes as like this, that is 3 and 4, if it is complementing 1 and 2 complementing, such a loop structure is coming, then that can influence the transcription. If this structure is coming for the mRNA which is produced from the 5' UTR region, then the first structure comes, then what happens, they can terminate the transcription. If the second structure 2 plus 3 secondary structure is coming, then the tryptophan level is low. The cell is sensing the tryptophan level is low. The region 2 and 3 pairs, this structure does not terminate the transcription. How it is happening? Why this is a sort of a mechanism? Already your regulatory protein is stopping the transcription. Then why you need another set of another set of control? Because the first set of control which is inhibiting the transcription 60%. You need additional 10%. So this mechanism is involved in this particular operon. See how this 5' UTR is influencing our transcription. How it is happening? That means you can see 2 tryptophan codon in the first region. Actually, if the RNA polymerase is after regulatory genes repressor come and bind with the operator also, then a small amount of transcription will happen in the cell. So what happens is, before to that, some of the RNA polymerase, maybe before binding with the operator region, a few RNA polymerase may be moving. Afterwards, sometimes this operator region is blocked. So the RNA polymerase might have reached region 1. So region 1's one's nucleotide before trip operon may be transcribing. When it reaches the trip codon, you need for the production of protein. For the production of proteins, this mRNA, when it's stalled with the ribosome, because transcription and translation in the prokaryote which is occurring side by side in the case of prokaryotes. So what happens when this particular trip code reaches near the trip codon, the mRNA will be plugged with or installed with the ribosome which need amino acid tRNA with the tryptophan. But the tryptophan level is already low. So what happens? It will stall there. It will stall there at the tryptophan codon to get the amino acid tRNA with the tryptophan. So what happens? In the first region, it will be stalled. In the first region of mRNA, it will be stalled. Then in meantime, what happens? The second and third part of mRNA, second and third part of mRNA will get looped, and to the fourth part of mRNA, it won't be that won't be coming into the ribosome, so that will be free and other part will be free. So that's such a structure you can see, such a peculiar 2+3 arrangement, because first you can see your 5'3' nucleotide. Nucleotidic region, you cannot get this type of arrangement. After transcription, during transcription itself, the mRNA will be stalled in the ribosome, and during translation time, each polypeptide is making polypeptide of protein is making by this translation process. During that process, when it reaches the trip codon of region 1, it will be stopped there due to lack of amino acid tRNA with the tryptophan has to be reached. Then only that part translation will occur. In meantime, the mRNA will achieve such an arrangement, 2+3 secondary structure they will form, and this structure formation is anti-termination because there is low amount of tryptophan. If a high amount of tryptophan is actually present and you want to stop them, then what happens? The cell having high amount of tryptophan, there is the amino acid tRNA with the tryptophan is not a big issue. That will come. And the transcription will proceed. Then this type of 1+2 and 3+4 secondary structure attenuation happens and termination occurs. This you can see in the next slide also. What happens? How this look here. Each mRNA is stalled with the ribosome, and first if it is low level of tryptophan, then the ribosome will be stalled here, and in the tryptophan operon without amino acid tRNA with the tryptophan. So the ribosome will be stalled, and the mRNA will get 2+3 arrangement, and this will enhance the structural gene transcription, and the tryptophan operon will happen in a regular fashion. They will transcribe. The RNA polymerase will easily move and go and bind with the structural protein transcription will proceed, and this mechanism occurs only at low level of tryptophan. If it is high level of tryptophan, there is no issue for the ribosome to translate all the part of mRNA. So they will proceed to second portion. Then what happens? 3 and 4 form a loop structure. This stem and loop arrangement signals the termination of transcription. Then what happens? The RNA polymerase will plucked off from the DNA sequence, and they cannot go and transcribe the structural genes. This is the actual mechanism of tryptophan operon. Actually, 2+3 arrangement which go and anti-terminate the transcription. If 3+4 arrangement comes, then what happens? That will terminate the transcription. This all about this topic. Thank you one and all for the kind listening.